Hi, I'm Wig for part 2 of protein synthesis. I first talked about the role of the cell organelles and then what a protein is and how it works in the cell and in the body. And now I want to talk about, we're going to start out with the 3 types of RNA that you find in protein synthesis. And we're going to be using this kit that you have made. So there's three different types of RNA. The first one we're going to talk about is this messenger RNA. And in your kit, it looks like this little part right here. Let me try to get this up. So in your kit, it looks just like this. So it'll say on there messenger RNA. And we'll actually make that messenger RNA from the DNA. So messenger RNA's job is to get the message. It gets the code from DNA and takes that message in the form of RNA from the nucleus to the ribosome and the cytoplasm. We're also going to see that every 3 nucleotides is a codon. So you can see AUG is one codon, GUG is a codon, because it's every 3 nucleotides. Another thing that I want you to notice is that DNA, we said, stays in the nucleus, and RNA, since DNA's bigger than RNA in that it's double-stranded and RNA is single-stranded. So RNA can actually get out of the nucleus. So messenger RNA is going to go out of the nucleus to the cytoplasm where the ribosome is found. This is going to illustrate the ribosome in our kit. Notice it has a small ribosomal subunit as well as a large ribosomal subunit. Another name for ribosomal RNA, which is made in the nucleus of the cell, is called the ribosome. So messenger RNA will attach to the ribosome as well as transfer RNA in the cytoplasm to make a protein in the final step of protein synthesis. The next RNA I want to introduce you to is transfer RNA. In your kit, it's going to look like this. It's going to have a UAC, for instance. That's going to be the anticodon that will pair hydrogen bond to the codon in messenger RNA. All transfer RNA will carry what we call an amino acid on top of it. In this case, UAC, that transfer RNA will carry MET. And the codon that will code for methionine or MET is going to be AUG, and it's going to be found on messenger RNA. That just so happens to be the start codon. So transfer RNA is going to hold the anticodon located right here, and it will base pair again with the codon found on messenger RNA. Transfer RNA is also going to hold our amino acid. So now that we know the 3 types of RNA, which are messenger RNA, ribosomal RNA, and transfer RNA, we're going to go into what is protein synthesis and why in the heck is Mrs. Hellwood making me learn about protein synthesis. Protein synthesis is the making of a protein. And I'm hoping that this model that I've made for you will help you understand how a protein's made. In this kit, we're going to look at normal red blood cells versus a sickle cell anemia, where the actual red blood cells actually sickle. Instead of being nice and round like a red blood cell, they're going to take on a sickle shape, which is a trait. So red blood cells are amazing because they start out and they can be any cell in the body. And then they decide to differentiate and become red blood cells. And when they do that, they'll literally spit out their nucleus, and so that they can just hold oxygen. And their job is going to be to hold oxygen and get oxygen to the rest of the body. So in our bodies, we have 20 different amino acids. And you can see that these amino acids are abbreviated by, for instance, here in this chart, phenylalanine is abbreviated PHE. And then we have this dictionary code of life, which is the code of life where messenger RNA, and I want to make sure you understand that messenger RNA will have the code to make these specific amino acids. So that if I have a codon on messenger RNA called AUG, how I would use this dictionary code of life is I'd first go to this side right here where it says first base, and I look for the A. So here you can see I found the A right here. That's the first base. Then I want to come up here to the top of the dictionary code of life and look for the second base. And if I'm looking for AUG, here's U. So I would then go down to my A that's found right here, AU, and I'd go over here to the right where it says the third base can be found. So I want to stay in that same vicinity where A is and U, and there it is. AUG is the MET. It codes for the amino acid MET or the start codon. So my messenger RNA will say AUG, and I'll use, here's where the first base is found. Second base will be found across the top, and the third base will be found across the right-hand side. So that's how you do it. So if we were looking for UUU on messenger RNA, we go here, first base is U, second base is U, come down here, all the way here to this U here on the third base, come over, and it just so happens that UUU codes for phenylalanine, another amino acid. So that's how you use the dictionary code of life, and we'll be looking at that later on in translation. So here you can do a little activity trying to see if you too understand what amino acid would code for AUG, what amino acid would code for GGC, and you can fill in the amino acids using the dictionary code of life. Now we're going to come down here and we're going to start protein synthesis. So the first part of protein synthesis is called transcription, and transcription is going to occur in the nucleus of the cell, and that's where we're going to copy DNA to messenger RNA. The second part of protein synthesis is messenger RNA processing, and sometimes they call it post-transcriptional processing. The location of that is going to be found in the nucleus of the cell, and messenger RNA, we're going to use messenger RNA, a 5' cap, which we call G3P, a poly A tail, which is just a lot of A's. They call that polyadenylation tail. And then what we're going to do there is we're going to also add, we're going to cut out the introns. So here we're going to cut the introns out with spliceosomes. So you'll see we'll need, in this process, we'll need a 5' cap, a poly A tail. We're going to need a spliceosome. We're going to cut the introns out. And the reason we need to put a 5' cap and a poly A tail on this is so that the journey from the nucleus to the cytosol has a lot of enzymes, a lot of things that can hurt messenger RNA by degradation or breaking down, like enzymes. And so we're going to put a cap and a tail on that so that it protects it. The last part in protein synthesis is called translation, and that's going to occur in the cytoplasm or cytosol of the cell, and that's where RNA is going to be actually translated to a protein. And what the players will look like in translation is we're going to see messenger RNA, we're going to see ribosomal RNA, we're going to see transfer RNA, we're going to see amino acids that get peptide bonded together to form polypeptides, which eventually will form a protein, and then a trait such as sickle cell or normal hemoglobin. So we're going to start out with this transcription. So I want you to know that we're located in the nucleus of the cell. So what you have to do is get out your DNA colored strip that I made for you, and it's going to start, it's a 3-section piece that you're going to have to tape together. So how you're going to tape it is, this is going to be strand 1 of 3. So on the left hand side you're going to put strand 1 of 3. It should show the TATA box right here, which is TA, and then 3 A's in orange. Then you're going to tape very closely together. I want you to butt these two together very closely. Tape strand 2 of 3 next to that, and then the final strand will, and again butt them very close together, that will be 3 of 3. So you should have 1 of 3, 2 of 3, and 3 of 3 all taped together in a nice linear sequence such as this. Once you get that taped together, I want you to draw a picture of the cell on your desk, and I want it to be the whole outline of the cell, like so. See how I went all the way down and drawn a great big cell. Then inside that cell we're going to draw a nucleus, And then inside that nucleus, we're going to place our DNA. So we're going to place that. You should also have something called a template strand. This DNA that is in nice colors here is called the coding strand or the sense strand if you have an older biology book. We're going to call it the coding strand. We need to copy the template strand by using the coding strand. So we're going to tape together the template strand the same way, 1 of 3, 2 of 3, 3 of 3, so that they will line up together and form a nice little DNA template. So it should look like this. So you can see where that TATA box right here, so that's going to be, if that's an A on the coding strand, it'll be a T on the template. So we're making a DNA template strand to complementary base pair with the coding strand. And note that if you have a T here and an A here, they're going to be held together by hydrogen bonds. T's and A's have 2 hydrogen bonds, and C's and G's always bond together, and they have 3 hydrogen bonds. So I should see only T's, A's, C's, and G's, because this is DNA. If this were RNA, I would be changing these T's to U's, because there are no U's or T's in RNA. So again, I want you to remember that each one of these bases that we see come with a sugar called deoxyribose sugar and 3 phosphates. 2 of the phosphates pop off and only 1 phosphate will adhere. So those form what we call a nucleotide, so that a phosphate, a deoxyribose sugar, and a nitrogen base. Again, there's 4 nitrogen bases in DNA: adenine, thymine, guanine, and cytosine, or A, T, C, and G.